Fantastic. Great to be here in Phoenix for another installment of Meeting on the Mesa. I'm Sean Ainsworth. I'm CEO of Immusoft. At Immusoft, we're developing B cells as a modality for sustained therapeutic protein delivery. More specifically, we're working with the subtype of B cell known as a plasma cell. It's an end-stage differentiated B cell that is really an extraordinary biofactory. There's a few key elements to B cells that have driven us to move in this direction. One of those being that these can be delivered without the need for any myeloablation, and they'll naturally migrate to the bone marrow, which once they engraft, they're producing therapeutic proteins to the tune of thousands per second. And so we're really excited to be in the clinic now. We're seeing great results in our first patient, both in terms of an extremely well-tolerated safety profile, but also quite remarkable efficacy, early signs of efficacy, but efficacy nonetheless in this trial. We're also very excited to announce that we recently redosed our first patient. So this is something that is actually just a huge impetus for Immusoft, and a big unifying theme on the team is to develop a modality that can be redosed. And to have achieved this now, we're very excited. And we've done this with, as I say, just a really extremely clean safety profile. The patient's doing exceptionally well. And I'll get into some of the data here briefly, but I think all of us in the room recognize that when we think about cell and gene therapies, we think about one and done, great, but what that means is you've got to get it right the first time. Most of the other modalities don't have the ability to go back and redose. So for us to be able to do that, has been really, really outstanding. And if we think about gene therapies, virus delivered in particular, getting that right often means that you're dosing on the order of trillions of virus particles, sometimes hundreds of trillions, up to a quadrillion. And when you do that, I think we all recognize that that bad things can happen. So we're in the clinic, we've dosed, we've redosed, and in terms of the safety profile, we've seen no related adverse events, no SAEs. The patients are doing outstanding. So a little bit about the B cells. A plasma cell, these are these extraordinary biofactories. In normal biology, they're producing and releasing thousands of antibodies per second into the circulation. And so we're programming those to produce a therapeutic protein of choice. In the case of our lead program, that protein is iduronidase. And so that is then being produced 24/7. Those B cells migrate to the bone marrow, where once engrafted, they can remain for years, potentially decades. And so a little bit about the way the approach works. We start with an apheresis. We actually don't need a mobilization step. We begin directly with the apheresis. We select out the B cells from the apheresis product. We program those ex vivo using a Sleeping Beauty transposon system. As I walk through these steps, all of them were prospectively designed to really, with scalability in mind. And so, just by way of example, that transposon system, that's naked DNA. So if we compare that against virus vectors, it's a very small fraction of the cost of virus vectors. And so the next step is to expand and differentiate those B cells. That's a 1-week process. We then administer them. They migrate to the bone marrow naturally, no need for a myeloablation regimen. And once they're in the bone marrow, they're producing those therapeutic proteins around the clock. And so when we talk about scalability, just by way of comparison, if we think about your traditional gene-modified stem cells, it starts actually with the mobilization regimen. That can sometimes lead to a hospitalization of up to a week. The manufacturing process then is usually a couple of weeks. that most of the time entails a lentivirus vector, which is also very expensive if I'm comparing against what we do. Again, we use that transposon system, naked DNA, very small fraction of the cost. We both have the release testing, but then in the case of the gene-modified stem cells, there's that myeloablation regimen that can lead to a hospitalization, usually 4 to 6 weeks. All of that gets baked into the cost of goods as well. So we avoid all of those hospitalization steps, the preconditioning, myeloablation. All of this really speaks to the scalability of our approach. Our lead indication is MPS I. MPS I is a lysosomal storage disease. It's characterized by the dysfunctional iduronidase enzyme. And so when that enzyme is not functioning properly, glycosaminoglycans, which are long-chain sugars, they don't get broken down properly. They build up in the cells, and at the cellular level, they're harmful, and so by extension through the tissues, organ systems. And so if these patients are not treated in the most severe form, the Hurler form, those patients won't live through childhood. Thankfully, there are treatments in the US and in the developed world, and we're really leveraging much of that in our lead program. And so in the case of the Hurler-Shea and Shea forms of MPS I, they're treated with an enzyme replacement therapy. And so we're leveraging much of that body of evidence with that enzyme replacement therapy that pretty substantially de-risks our program from a technology perspective. And so being in the clinic now, we've dosed a couple of patients. We've also had a successful Type C meeting with the FDA recently, gotten a lot of clarity on what the requirements to go from here through to approval are. That includes a single-arm study, which we're really excited about, as well as we've got an agreement on the registrable endpoints, the size of the study, and agreement to move into pediatric patients. Our clinical trial sites are University of California, UCSF, and University of Minnesota, and Drs. Paul Orchard, Paul Harmatz. They're really two of the leading key opinion leaders in the world. If we look at that safety profile, as I mentioned, we've seen nothing in the way of adverse events, nothing related, no SAEs. And if we contrast that against gene-modified stem cells that require that myeloablation regimen, or if we contrast that against virus, which requires that immunosuppression regimen, there's really no comparison. Our approach has shown a very favorable profile. We've also seen incredible signs of efficacy. And so this is a 6-minute walk test. The patient at the outset, at baseline, his distance was what you would expect really from about an 85-year-old man. And so if we fast forward to a year post-dosing, he's seen a 240-meter improvement. For those of you who may not know, usually about a 30-meter improvement is considered clinically meaningful. And so in the case of this first patient, he's 25 years old, and he's now in the normal range for his age. So we've seen really remarkable improvements across many fronts. Without going into the details of all the data, I'll speak a little bit to the pharmacodynamics. We've seen decreases in those GAGs in the urine, which is, we've normalized those. We've seen those functional improvements in addition to the 6-minute walk test, improvements in shoulder flexion, which is a big problem in this patient population. And then in a way of patient-reported outcomes, among other things, he's reported a pretty substantial reduction in pain. And most of these patients are actually taking ibuprofen around the clock to manage pain, and our patient is essentially discontinued. And at his last visit, in fact, he reported no pain across an 11-question pain questionnaire. So I don't think I can report that myself. Nonetheless, very excited by that. And what all that really adds up to is validation, I think, for this platform more broadly speaking. And so when we think about the commercial opportunity, I want to give a thanks to Alliance for Regenerative Medicine. We really liked this slide and borrowed it as a result. And I think I'm probably preaching to the choir here, but it's important to remember that in terms of cell and gene therapies, there's a much greater likelihood of getting to approval than with with other modalities. In the case of these advanced therapeutics, there's also shorter timelines, much more manageable clinical trial sizes, and last but not least, the cost of goods is coming down year over year, and that's part of the equation for us when we contemplate scalability as well. So our strategy has been to start with MPS I. As mentioned, it's genetically defined. There's already a therapeutic protein on the market that's been de-risked, and that is, for all intents and purposes, what we're delivering in our lead program. And so we deliberately took a step to MPS II as a closely related indication. And the rationale for that is to talk to the FDA about platform consideration. And we were successful in doing that. So the FDA has reviewed what we've done with MPS I. They're looking at what we've done for MPS II, and the burden to get into the clinic for MPS II is much, much lower. And so as we start to think about that across the platform more broadly, that can save a year, maybe more per program, and potentially millions of dollars. So from here, we've got other larger indications that we've been doing some work on, a lot of partnering interest in those indications. Those are higher risk, higher reward, but I think some of the big pharmas really have that appetite, so that's encouraging and very exciting. And then Gaucher is a related lysosomal disorder that we're also getting some really compelling early data in. So as we look at our pipeline more broadly speaking, firstly, I want to say thanks to CIRM. They've been outstanding for supporting us in both our MPS I and MPS II programs. I want to acknowledge ARPA-H as well. So collectively, I think we've gotten $26 million in non-dilutive financing from those institutions. And so we're, as you can see, beginning with the genetically defined approaches, moving from there into the larger therapeutic areas. This is really an extraordinary team to work with. We've been able to do an incredible amount of work with a relatively small team, and it's because it's just everybody is focused on the end goal in mind, getting these advanced therapeutics into patients safely, effectively. Across the board, we've got experience in big pharmas, small biotechs, taking programs into the clinic, taking them through to approval. Our intellectual property actually started at Caltech with David Baltimore's lab. They were the first to conceptualize and reduce to practice engineering B cells to deliver bespoke therapeutic proteins. So we've licensed that intellectual property and built on that, and collectively we've got a dominant intellectual property position as well as freedom to operate. And so, we're extraordinarily excited about what the future holds for us. We think that, this approach is going to change the way that we conceptualize cell and gene therapies and advanced therapeutics. And, I look at this engineered B cell approach as really an advanced therapeutic, a next generation advanced therapeutic. And so would welcome a chance to talk to those who might be interested in learning more. Thank you very much.